Good morning, everybody. This morning's lecture is going to be about Mendelian inheritance, and let's look at what Mendelian inheritance means. Mendelian inheritance refers to patterns of inheritance observed in traits governed by a single gene. Gregor Mendel was the father of modern genetics, and he first described the principles of Mendelian inheritance. This presentation will explore into the key principles and examples of Mendelian inheritance. Let's look at Mendel's laws of inheritance. And the first law is the law of segregation. And the law of segregation states that different genes and their alleles are inherited within sexually reproductive organs. Or during the formation of gametes, the alleles or the forms of gene separate with one allele from each parent, passing to the offspring. This is during the process of meiosis. If you still remember the process of cell division by the process of meiosis, when gametes are forming, the alleles or the different forms of gene separate with one allele from each parent, passing it to the offspring. Which means that each individual possesses two alleles for a given trait, but only one allele is passed on to each offspring. If you look at the diagram below, it shows the maternal and the paternal alleles segregating during the time of meiosis, and when the maternal and paternal alleles have segregated into the separate daughter cells or gametes. The second law of Mendel is the law of independent assortment. According to his law of independent assortment, genes for different traits segregate independently of one another during gamete formation. This law applies to gametes located on different chromosomes or on the same chromosome far apart. When we talk of genes from different traits segregating, different traits could mean genes that portray the size of the individual, the color, the height. So the diagram below is trying to depict homologous chromosomes, and these homologous chromosomes are crossing over, or they have crossed over. So when they cross over, it means that there is going to be a recombination of the chromatids, a recombination of the chromosomes. So when these chromosomes recombine, it means that this will bring about the production of different traits in an individual. Let's continue with the Mendelian inheritance. Crossing over between homologous chromosomes occurs between non-sister chromatids of homologous chromosomes, as in the previous diagram. So the result will be an exchange of genetic material between the homologous chromosomes, and this will lead into two recombinant chromosomes and the two non-recombinant chromosomes. Let's look at the types of Mendelian inheritance. Number one, we have dominant inheritance. In dominant inheritance, a single copy of a dominant allele is sufficient to express the trait. When we talk of a trait could mean different characteristics or different entities that are inherited from the parents to the offspring. It could be the skin color, the hair color, the blood group, and so on. So the dominant allele masks the presence of recessive allele. Do not forget, allele means a variant of a gene. So examples of dominant allele include Huntington's disease and polydactyly, that is extra fingers or toes. Let's continue with the types of Mendelian inheritance. Codominant inheritance. In codominant inheritance, both alleles contribute to the phenotype, resulting in a combined expression. Neither allele is dominant or recessive over the other. Examples of this include the ABO blood group system, where A and B alleles are codominant. The diagram below is depicting codominance. For example, the blood type. Let's look at Punnett squares and genetic process. Punnett squares are tools used to predict the possible outcomes of genetic crosses. They represent the combination of alleles from both parents and the resulting genotypes and phenotypes of offspring. By using the principles of the Mendelian principle, Punnett squares can determine the probability of specific traits or disorders in offspring. For example, the diagram below is showing a male and a female, where we have the A as being the dominant gene and the A being the recessive gene. So when there is crossing over that takes place, it means that when A crosses over with A, it means that AA will be dominant. Then we have we have capital AA, then we have capital A, small a. So the ratios of the phenotype will be 3 to 1. Let's look at examples of Mendelian inheritance. Example number 1 is autosomal dominant. For example, we have the Mendelian syndrome. Mendelian syndrome is a connective tissue disorder characterized by long bones, aortic abnormalities, and vision problems. We also have achondroplasia. Achondroplasia refers to dwarfism resulting in short stature and abnormal bone growth. The diagram below depicts the inheritance, depicts autosomal dominant inheritance, whereby we have the father who is carrying the mutated gene and the normal gene. Then we have it means the father is affected because he is carrying the mutated gene and normal gene. Then we have the unaffected mother who is having two genes that are normal. So their offspring, one of their daughters will be an affected daughter. Then the second son will be an affected son. Then the other daughter will be an affected daughter. Then the fourth child will be an affected son. That is when the crossing over of the chromosomes occur and the inheritance the genes. Let's look at autosomal recessive. One of the autosomal recessive, an example is albinism. Albinism refers to lack of pigment in the skin, hair, and eyes. This is because of a mutation in genes that are involved in melanin production. Then we have the Tay-Sachs disease. This is a neurodegenerative disorder causing progressive deterioration of motor and mental abilities. Below we have a diagram depicting the inheritance from the parents, whereby we have an unaffected carrier who is the father, then we have an affected carrier who is the mother. It means that the part which is shown in blue, the genes are normal. Then the part which is shown in orange, the genes are abnormal. So their offspring will be, they will have one of the first child, he will be not affected. The second child will be unaffected but a carrier child. Then the third child will be unaffected carrier child. Then the third one will be affected child. Let's look at pedigree analysis. Pedigree analysis is a visual representation of inheritance patterns within a family. The pedigree analysis helps to track the occurrence of traits or disorders across multiple generations. Pedigrees can provide insights into mode of inheritance, whether it is autosomal dominant, autosomal recessive, or X-linked, and aid in genetic counseling. Let's look at the non-Mendelian patterns in humans. While Mendelian inheritance describes the patterns of inheritance for single gene traits, there are instances also where inheritance patterns deviate from these principles. Non-Mendelian patterns involve more complex genetic interactions, including incomplete dominance, codominance, polygenic inheritance, and sex-linked inheritance. This presentation will explore the key non-Mendelian patterns observed in human genetics. Let's look at incomplete dominance. In incomplete dominance, neither allele is completely dominant, resulting into an intermediate phenotype. The heterozygous genotype exhibits or shows a phenotype that blends the two homozygous genotypes. An example is an inheritance of flower color in snap-growns where red and white alleles resulted in two pink flowers when inherited together. Let's look at codominance. In codominance, both alleles are fully expressed in the heterozygous genotype, resulting in simultaneous presence of both phenotypes. Here, there is no blending of traits, but rather the expression of both traits side by side. A classic example is the ABO blood group system, where both A and B alleles are codominant, resulting in two AB blood types. Let's look at polygenic inheritance. Polygenic inheritance involves the interaction of multiple genes to determine a single trait. The phenotype is influenced by cumulative effect of multiple genes, resulting in a continuous range of phenotypic variations. Examples of this include the height, skin color, intelligence, where multiple genes contributed to the observed variations in the population. Let's look at the sex-linked inheritance. Sex-linked inheritance involves inheriting traits or disorders carried on the sex chromosome, that is X and Y. Certain traits are more commonly observed in one sex due to their presence on the sex chromosomes. The X-linked inheritance is more common, with examples such as color blindness, hemophilia, where the gene is located on X chromosomes, and color blindness mostly affects the male. Let's look at mitochondrial inheritance. Mitochondrial inheritance involves the inheritance of genetic material exclusively from the mother through the mitochondria. Mitochondria have their own DNA separate from the nuclear DNA, and mitochondrial DNA are passed on to the offspring through the cytoplasm of the egg. Diseases associated with mitochondrial inheritance include Leigh syndrome and the Leber's hereditary optic neuropathy. Let's look at complex genetic interactions. In addition to the mentioned non-Mendelian patterns, complex genetic interactions can involve combinations of different modes of inheritance. For example, epistaxis, whereby one gene affects or masks the expression of another gene, is an example of complex genetic interaction. So understanding these complex interactions is very important for unraveling the genetic basis of various traits and disorders. Let's look at genetic disorders. Genetic disorders are conditions caused by abnormalities or mutations in an individual's gene or chromosomes. These disorders can be inherited from parents or they can occur as simultaneous mutations. Let's look at chromosomal abnormalities. Chromosomal abnormalities involve changes in the structure and number of chromosomes. Examples include the Down syndrome or trisomy 21, where there is an extra copy of chromosome 21. And if there is an extra copy of chromosome 21, this leads to intellectual disability and also certain physiological features. Another example is Turner's syndrome. In Turner's syndrome, there is a missing or a partially deleted X chromosome in females. So individuals who have Turner's syndrome usually are short in stature and they are also infertile. Third example is Klinefelter syndrome. In Klinefelter syndrome, there is an extra X chromosome in the males, whereby the male is now considered to be XXY, and individuals who possess Klinefelter syndrome, they are usually infertile and also have developmental delays. Let's look at single gene defects. Single gene defects, these are mutations or alterations that cause single gene defects in a single gene. Let's look, the types of single gene defects include the following. We have the autosomal dominant disorders, the autosomal dominant disorders. These are mutations in a single copy of a gene on an autosomal chromosome, for example, Huntington's disease and Mendelian syndrome. Then we have the single cell gene defects. These are autorecessive disorders, and these autorecessive disorders are mutations in both copies of a gene on an autosomal chromosome, for example, cystic fibrosis and sickle cell disease. Then we have the X-linked disorders. These are mutations in genes located on the X chromosome. For example, we have the Duchenne's muscular dystrophy and hemophilia. Let's look at multifactorial diseases. Multifactorial diseases result from the combined effect of multiple genetic and environmental factors. These disorders have an extra inheritance pattern and are influenced by the genetic predisposition and environmental triggerers. Some of these diseases include heart disease, diabetes, and schizophrenia. Let's look at cancer. Cancer is a genetic disorder characterized by uncontrolled cell growth and division. It is often caused by mutations or alterations in specific genes involved in cell cycle regulation and tumor suppression. Let's look at the types of genetic alterations that can lead to cancer. One of it is oncogenes. Oncogenes are mutated genes that promote cell growth and division. We have the tumor suppressor genes. These are genes that normally regulate cell growth and prevent tumor formation. Then we have the DNA repair genes. DNA repair genes, these are genes involved in repairing DNA. And if they are damaged, it means that there is no maintenance of the genomic stability, and this can lead to cancer. Let's look at the causes of genetic disorders. Inherited mutations passed down from the parents. This can lead to genetic disorder. Spontaneous mutations, this can occur during gamete formation or early development. Environmental factors such as exposure to toxins or radiation, this can damage the DNA. Then gene-environment interactions, where genetic susceptibility interacts with environmental factors to trigger disease. Let's look at genetic testing and counseling. Genetic testing plays a crucial role in diagnosing and managing genetic disorders. Genetic testing involves analyzing the individual's DNA to identify genetic variants or mutations associated with specific disorders. So genetic counseling provides support and guidance to individuals and families regarding implications of genetic disorders, inherited patterns, and available options for management and prevention. Let's look at advancements in genetic research and medical interventions. Genetic research has led to significant advancements in understanding human genetics and its applications in healthcare. So techniques have been used such as gene therapy, genetic testing, personalized medicine have also revolutionized medical intervention. So these achievements hold promise for targeted treatments and improved patient outcomes. Let's look at ethical considerations in human genetics. As our understanding of human genetics expands, ethical considerations become increasingly important. Issues such as privacy, informed consent, genetic discrimination, and the equitable access to genetic services need to be addressed. Therefore, balancing knowledge and responsibility ensures that genetic advancements are used ethically and responsibly for the benefits of the individuals and the society. In summary, we define Mendelian inheritance and its laws. We looked at the types of inheritance and its examples. We looked at pedigree analysis, non-Mendelian patterns of inheritance for single gene traits, and we also looked at various examples. We looked at the sex-linked inheritance, genetic disorders which included chromosomal abnormalities. We also looked at advancements in genetic research, medical interactions, and ethical counseling in human genetics. Thank you for listening and have a blessed day.